All right, hello biology students. We are doing a lab today. We actually started it yesterday, but we thought we would just give you the background knowledge about what we did, and then we'll fill you in on what we're doing today. So yesterday we took some pineapple. So we had some chopped up pineapple, and we had another, I already threw it away, but we had a can of canned pineapple. So we had these two groups of pineapple which have inside of them an enzyme, a certain enzyme that allows the pineapple to break up some protein and use the little bits of protein for its own use. So it's called bromelain. So this enzyme is something that would break up proteins. So what we wanted to see is what would happen if we allowed that enzyme to do its job under different temperatures. So we wanted to see what kind of relationship there is between temperature and enzyme activity. So one important thing that you might need to have for background knowledge is that when you put pineapple in a can and you heat it up, in order to can the pineapple, you have to heat it up. So once it's gone through that process, it's been heated up so much that the protein, the enzyme, it gets warped out of shape because it gets so hot while it's canning. And so the warped, changed shape of the enzyme makes it so it doesn't work anymore. So we're going to do two experiments really. One big experiment is going to see how the temperature will affect the enzyme activity, but the other one is going to see if it's been canned, does the enzyme work at all in that situation. So if it's been warped, will the enzyme still work? So we basically have two experiments that we're doing at once. Okay, so what we did is we made these cups of gelatin, and we let them set overnight so they were solid. And gelatin is made out of protein, so when we add pineapple to it, those enzymes will start to chop up the proteins in the gelatin and make it liquid again. So we blended up some pineapple, we blended up some canned and fresh pineapple, and then added it to cups. So a whole group of cups, half of them got fresh, half of them got canned. And so we put them in different temperatures. So one group was kept in the refrigerator overnight. That was 10 degrees Celsius. Another group was kept in room temperature overnight, which was, I think, 22 degrees Celsius. And then one group was kept in a heating oven, which was 29 degrees Celsius, which is pretty warm. It's like 80 degrees, 85 degrees Fahrenheit. And we left them overnight. And we took them out, and so here we are. We're going to take a look to see how much liquid is produced. They all started as solids, and so we want to see how much activity the enzymes had. So we're going to show you how we're going to make those measurements, and then we'll give you the data afterward. And so I'll start with the cold temperature. So let's see. I'll start with the fresh pineapple cold. That's how we labeled it. And so remember, this started with solid gelatin, and we added some fresh pineapple to it, but it's been in the cold temperature the whole time. So whatever liquid's produced, that tells us how much enzyme activity there's been. So we'll see how much enzyme activity there's been in cold temperatures. So I would pour it over this wire grate, just because if anything's solid, then it'll catch it, and all the liquid will drip through. As you can see, you don't have very much at all. So I'm going to measure how much liquid that is. We have a grand total of 0.5 milliliters for that trial. 0.5. So I'm going to write that down. That was for fresh pineapple in cold temperatures. 0.5 milliliters. And we're going to do the others off camera so you don't have to watch that over and over again. Okay, so we'll do that again with the, I did fresh, so this is the canned pineapple in the cold temperature. See if there's any difference. Everything else stayed in the cup. Pretty much 0. I guess I have a little drop there. I'll measure that. So I'll use this. Yeah, okay, about half of a milliliter. So I'm gonna put that in my data table here. That was canned pineapple and cold. There we go. Okay, so that would be 2. So I'll put all my waste liquid in there. You know, what would happen if I didn't get all the liquid out of these graduated cylinders? Then my numbers would be different, wouldn't they? So that's actually a little detail, but pretty important detail, that I make sure that when I measure my volume, that I'm only measuring the liquid from each trial instead of residual liquid each time. Okay, now I'm going to move on to the room temperature. So I'm going to do the same thing. This is fresh pineapple and room temperature. We got a lot more liquid here. Now, an important thing to note is that we did everything exactly the same. We used the same water, we used the same cups, you know, we used the same blenders. Everything was kept the same except for the temperature they were exposed in and the type of pineapple that we used, canned or fresh. So that's an important thing to be thinking about when we designed our experiment. We wanted it to be a controlled experiment. So the only variables that were changed were the manipulated variables, which in our case is the temperature and the type of pineapple. Okay, all right, so this is room temperature, and it was fresh, and we've got 7.5 milliliters in this one. Room temperature and fresh, so 7.5 milliliters for that trial. You have a guest here, Mr. Ricky. This is a great biology team we've got here, by the way. Okay, so then we're gonna do the same thing with the canned pineapple in room temperature, so that'd be this group here. All right, so this is where it gets interesting. We'll start seeing any differences between these groups. Okay, so I don't have any liquid in there. We'll see how much liquid comes out of the canned pineapple. Okay, look like a whole lot here. Okay, got a few drips coming out. You can measure how much volume that was. Use this graduated cylinder, it's empty. That's only a few drips, really. Okay, my volume here is 0.5 milliliters. 0.5, that was for canned pineapple in room temperature. Notice we're starting to get some differences in the canned versus the fresh. Remember what I told you about how the canning process might warp that enzyme and keep it from working correctly? Okay, now we're going to move on into the warm temperature. And by the way, pineapple, you know a little bit about pineapple, don't you? Where it grows, tropical places. What temperatures do you think would be the best temperatures for an enzyme that is found naturally in a pineapple? Well, we're gonna find out. Okay, so we got fresh, warm, and I'm gonna measure the liquid that comes out of this one. Remember, everything's been kept exactly the same in all these trials, except for the temperature. I'm gonna need to use a bigger graduated cylinder to measure that. Got one over here. Okay, we'll see how much liquid got produced in our fresh pineapple warm. Remember, I started with 50 milliliters of gelatin. Now I have 50 milliliters of liquid gelatin. 50. So that was fresh pineapple in the warm temperatures. Okay, one last trial. We're gonna try the canned pineapple in the warm. Remember, the only difference between that group and this is that we used canned pineapple instead of fresh pineapple. They're both kept in the same place, nice warm temperatures. Okay, well, got some liquid here. Some of it didn't quite go in that beaker. I'll try to collect it afterward. Okay. So that's like semi-solid. This is why we're using that filter, because it's hard to tell at this stage whether something's solid or liquid. So we're gonna call the rest of that, solid. Guess I will try to collect up that liquid there. Okay. There, I got most of it. It's just a little bit on the counter there. One of the things you're going to be asked about in this lab is what is a source of error, meaning like what could have explained why our data might have been differently, might have been different than what we would have expected. And I do notice that some of my liquid was left on the table, just slightly. Okay, so semi-solid, semi-liquid. Okay, I've got a grand total of, well, I got 10 in this, a little bit more than 10 there. Okay, I have 10 there. Probably should use the big graduated cylinder. We have a little bit more. There we go. Here's the last little bit. Okay. Now, hopefully I got all of the liquid out of this beaker. Some of it's still stuck on the side. Tried to get most of it out, but our measurement might not have included some of the little bits that got stuck on the inside of the beaker or got left on the table, right? So that's always gonna happen in science. We're gonna have measurements that are not perfectly accurate. Okay, so actually this is a little bit more than 10, so I have to transfer some over so I can add the numbers together. My graduated cylinder only goes up to 10. So I have 9.6, and I have 3.6, 9, 10, 11, 12, 13.2. 13.2 milliliters. Again, I should have just used the larger graduated cylinder and I wouldn't have had to add those two numbers together. Okay, so we've got some data. We're gonna do the rest of it so you don't have to keep watching us do that for two more. We did 3 cups of each type. Why? You know, we have numbers here, but the more numbers we have, the more certain we're gonna be that our results actually are meaningful and that they're reliable, so that if we did this experiment again, we get the same results again. So having more trials gives us more confidence in our results. So we have 2 more sets of cups we're gonna measure, and we're gonna give you all the data afterward and then have you take a look at it after that. Okay.